Assalamualaikum and hello everyone, and welcome to our lecture on genitourinary tract infection. Today, we'll be exploring the fascinating world of microorganisms that inhabit and sometimes infect our urinary and reproductive systems. This knowledge is crucial for your future work in medical laboratories, so let's dive right in. Let's start by understanding what's normally present in a healthy genitourinary tract. In the urethra, the tube that carries urine out of the body, we generally expect to find very few microorganisms. It's typically considered sterile, especially in its upper portions. However, near the opening, you might find some bacteria that have made their way up from the skin. The vagina, on the other hand, is home to a rich community of microorganisms, dominated by lactobacillus species. These friendly bacteria are incredibly important. They produce lactic acid, which maintains an acidic environment in the vagina. This acidity acts as a natural defense mechanism, inhibiting the growth of many harmful microorganisms. For our male colleagues, the external genitalia have a relatively sparse microbiota, mainly consisting of typical skin flora like Staphylococcus and Corynebacterium species. Understanding this normal flora is crucial because changes in these microbial communities can often be the first sign of an infection or imbalance. Now, let's move on to urinary tract infections, commonly known as UTIs. These are among the most common bacterial infections affecting millions of people each year. The most frequent culprit behind UTIs is a bacteria you'll become very familiar with in your career: Escherichia coli, or E. coli for short. This bacterium is responsible for about 80% of all UTIs. But why is E. coli so common? Well, it's normally found in our intestines, and due to the close proximity of the urethra to the anus, especially in women, it can easily travel and colonize the urinary tract. Speaking of women, they are at a higher risk of developing UTIs compared to men. This is primarily due to their shorter urethra, which provides an easier path for bacteria to reach the bladder. Other risk factors include sexual activity, which can introduce bacteria into the urethra, and the use of certain types of birth control, like diaphragms or spermicides. Catheterization is another significant risk factor. When a catheter is inserted into the bladder, it can introduce bacteria or provide a surface for bacteria to grow on. This is why proper catheter care is so crucial in healthcare settings. While E. coli is the most common cause, other bacteria can cause UTIs too. These include Klebsiella pneumonia, Proteus mirabilis, Enterococcus faecalis, and Staphylococcus saprophyticus. This last one, S. saprophyticus, is particularly common in young, sexually active women. When it comes to diagnosing UTIs, we have several tools at our disposal. A simple urine dipstick can detect leukocyte esterase, an enzyme released by white blood cells, and nitrites produced by many UTI-causing bacteria, giving us a quick indication of a possible infection. Microscopy is another valuable tool. We look for the presence of white blood cells and bacteria in the urine. However, the gold standard for diagnosis is still the good old urine culture. This allows us to identify the specific bacteria causing the infection and determine which antibiotics will be most effective. Moving on to a related but distinct topic, let's discuss sexually transmitted infections, or STIs. These are infections that are primarily spread through sexual contact, and they can be caused by bacteria, viruses, or other microorganisms. Let's start with bacterial STIs. One of the most well-known is gonorrhea, caused by Neisseria gonorrhoeae. These are gram-negative diplococci, which means they're bacteria that appear in pairs and turn pink in a gram stain. In the lab, we can diagnose gonorrhea through a gram stain of urethral discharge, culture on selective media like Thayer-Martin agar, or through nucleic acid amplification tests, also known as NATs. Another important bacterial STI is chlamydia, caused by Chlamydia trachomatis. This is a tricky one because it's an obligate intracellular bacterium, meaning it can only grow inside host cells. It's also often asymptomatic, which makes it easy to spread unknowingly. For diagnosis, we primarily rely on NETs, or less commonly, cell culture. The third bacterial STI we'll discuss is syphilis, caused by Treponema pallidum. This spirochete bacterium causes a multi-stage disease that can have serious long-term consequences if left untreated. Diagnosis typically involves serological tests like RPR, which stands for Rapid Plasma Reagin, VDRL, or Venereal Disease Research Laboratory, and TPHA, or Treponema pallidum hemagglutination assay. Now, let's move on to viral STIs. First up is human papillomavirus, or HPV. This DNA virus is incredibly common and can cause genital warts and several types of cancer, including cervical cancer. We can detect HPV through PCR tests or indirectly through abnormal puppy smear results. Herpes simplex virus, both HSV1 and HSV2, can cause genital herpes. These viruses establish lifelong infections with periodic outbreaks. We can diagnose herpes through PCR, viral culture, or serology. Lastly, but certainly not least, is human immunodeficiency virus, or HIV. This retrovirus attacks the immune system and, if left untreated, leads to AIDS. HIV testing typically involves an initial screening test like ELISA, followed by confirmatory tests like Western blot or PCR. There are also some other important genital pathogens to be aware of. Trichomonas vaginalis is a protozoan parasite that causes trichomoniasis. We can often spot its characteristic tumbling motility under the microscope in a wet mount preparation. Finally, let's not forget about Candida albicans, a yeast that can cause vulvovaginal candidiasis, often referred to as a yeast infection. We can identify Candida through microscopy or culture. Now, all of our diagnostic prowess wouldn't mean much if we don't collect and handle specimens properly. For urinary tract infections, we typically use a clean catch midstream urine sample. This means the patient cleans the genital area, starts urinating, and then collects the middle portion of the urine stream in a sterile container. This helps to avoid contamination from the skin. For sexually transmitted infections, we might collect urethral, cervical, or vaginal swabs, depending on the suspected pathogen and the patient's anatomy. It's crucial to use the right type of swab and transport medium for each test. Proper labeling is absolutely critical. Each specimen should be clearly labeled with the patient's information, date, and time of collection, and the type of specimen. Time is also of the essence. Many of these pathogens are fragile and won't survive long outside the body. Specimens should be transported to the lab and processed as quickly as possible to prevent overgrowth of contaminants and death of pathogens. Once we get the specimen to the lab, we have several techniques at our disposal. Microscopy is often our first step. We might do a wet mount preparation to look for motile trichomonids or yeast. Gram staining can help us identify bacterial morphology and provide a quick presumptive diagnosis in some cases. For some pathogens, like chlamydia, we might use fluorescence microscopy. Culture methods are still a mainstay of diagnosis. We use selective and differential media to isolate and identify specific pathogens. For example, we might use Thayer-Martin agar for N. gonorrhoeae or Saburot agar for Candida. Don't forget about anaerobic culture techniques for organisms that can't tolerate oxygen. Biochemical tests help us identify bacteria based on their metabolic capabilities. We might use API strips or automated systems like Vitek for this. Increasingly, we're using molecular diagnostic methods. PCR and other nucleic acid amplification tests can detect pathogens quickly and with high sensitivity. These are especially useful for difficult to culture organisms like chlamydia and viruses. For bacterial infections, determining antibiotic susceptibility is crucial. We have several methods for this. The disc diffusion method involves placing antibiotic-impregnated discs on an agar plate inoculated with the bacteria and measuring the zone of inhibition. Broth microdilution allows us to determine the minimum inhibitory concentration, or MIC, of an antibiotic. The e-test is a convenient method that combines principles of both disc diffusion and broth dilution. Many labs now use automated systems for both identification and susceptibility testing. Finally, interpreting and reporting results accurately is crucial. We need to understand the clinical context. For example, the presence of bacteria in urine doesn't always indicate an infection in catheterized patients. We must differentiate between colonization and true infection. This often requires correlation with clinical symptoms and white blood cell counts. Our reports should be clear and concise, providing clinically relevant information. And remember, if we find something significant or unusual, it's our responsibility to communicate directly with the healthcare provider. And there you have it. We've covered a lot of ground today, from the normal flora of the genitourinary tract to the various pathogens that can infect it, and the laboratory techniques we use to identify these infections. Remember, as future medical laboratory professionals, your work will be crucial in diagnosing and managing these infections. The accuracy of your results can significantly impact patient care.